For today's webinar, we have Dr. Glenn Jackson from the Department of Forensic and Investigative Science at West Virginia University, presenting Structural Characterization of Emerging Synthetic Drugs by Mass Spectrometry. Without further delay, here's Dr. Jackson.
So in the seminar today, I'd like to cover three main topics that we've been covering for the past 2 or 3 years as part of this NIJ-funded grant. The first third of the talk basically will be on the structural characterization of cathinones. We'll be looking at the tandem mass spectra, mostly of protonated cathinones and how they fragment in different types of mass spectrometers, and how we can use the fragmentation patterns to determine something about the structure of those cathinones. We've discovered a new tropylium, the pathway to the tropylium ion through these phthalate intermediates, and we'll be talking all about that. The second third of the talk, we'll be talking about fentanyl analogs and the identification of a third nominal isobar, MOZ188. MOZ188 tends to be the most abundant fragment ion in the tandem mass spectra of protonated phentalogs. And so we think it's quite important to have identified a third structure there. We've also identified a novel R group transfer that we'll talk about, and the effect of different modifications on the fragmentation patterns. And finally, we'll talk about something related but slightly different, and that is a new algorithm to assist practitioners with structural identifications. And the goal here is basically to help practitioners have some kind of probability or some kind of error rate related to a spectral comparison. So let's get started with the cathinones. Given the audience, I probably don't have to spend much time telling you about the importance of cathinones, but what we did here was use a variety of mass spectrometric techniques. We used tandem mass spectrometry and multiple stage mass spectrometry, so MS to the 2, MS to the 3, and MS to the 4, using ion traps. We used high resolution mass spectrometry using QTOF instruments, and isotope labeling studies with some custom synthesized cathinones by our collaborators at Auburn University. We used density functional theory calculations with collaborators at in Northern Illinois, and we used ion spectroscopy as well in the Netherlands at the Felix spectroscopy facility there. So again, synthetic cathinones basically derived from cat originally. These are the benzoyl ethanamine type compounds as shown in the bottom here. And the point is that they are very readily modified to provide a variety of different psychoactive effects. They can be swallowed, smoked, snorted, or injected, and they are a problem in society, both because people overdose and because they tend to behave in very unpredictable ways when they're under the influence. So let's get started with the tropylium ion. We got started on this project because in seminar here in our Department of Forensic and Investigative Science at WVU, there was a graduate student, a first-year master's student, giving a presentation, kind of a literature talk, about the first observation of cathinones in the literature, these like novel psychoactive substances based on cathinones in the literature. And the student showed this tandem mass spectrum. This is of protonated alpha PVP or FLACA. And what you can see here is the most abundant peak in this fragmentation pattern is MOZ91. And it was basically stated, and it's very well known, that MOZ91 is more than likely a tropylium ion. It's C7H7, and it's one of the most common fragment ions in EI mass spectra of anything that contains an aromatic ring. Well, so when I say anything, it has to have a couple of carbons and hydrogens available as well, of course. And my question really here was, well, this is odd, because if you look at the structure in the first place, you have a carbonyl group adjacent to that aromatic ring, which means that you have 6 carbons on the aromatic ring, and then the 7th carbon has an oxygen but no hydrogens. And then if you go to the next carbon over, the alpha carbon on which the pyrrolidine ring is bound, then there's only 1 hydrogen there too. And so if you think about the need to get C7H7, there are 6 carbons here and 5 hydrogens available, but it's really hard to imagine how you get a 7th carbon and 2 hydrogens. And this particular publication made no mention of how you get to the tropylium ion whatsoever. So we did a literature research and basically found that this was a very common observation for cathinones of all kinds. This is this is PV8, so a slightly longer alkyl chain here. And in this particular case, again, the base peak is M over Z91. In this case, they had high mass accuracy, and they were able to show that it is in fact C7H7 and consistent with the tropylium ion. And again, this is just showing that, how on earth do you get there? The only thing Swartwood's group, Marilyn Husis's group, actually mentioned here was that just that the most intense fragment was consistent with tropylium. But again, they made no mention of how you get there. So we went to the literature some more, and we found many more examples, actually, where all the examples we can find of tandem mass spectra of all the variety of different functionalized cathinones, whether they're functionalized on the aromatic ring out here for methyl dimethyls, methylene dioxy groups, etc. We found as many tandem mass spectra as we could, and we did find some general rules. And the rules are like this. If you have a good leaving group, like a pyrrolidine ring, then you tended to see this tropylium ion or substituted tropylium ion. And if you had a long alkyl group out here, then you tended to see it also. And it didn't really matter if you were functionalized on the aromatic ring, that functional group remained on the effective tropylium ion. And so the base peak would shift relative to tropylium at M over Z91. It would shift to whatever the additional mass of the functional group was. And just to give you a couple of examples, here's an example of a methoxy substituted cathinone. You can see that the base peak here is M over Z21, which is shifted by the mass of the methoxy group relative to just the pure tropylium ion. And again, here is the tandem mass spectrum of 4-hydroxy alpha PVP. So it's just a hydroxy group down here. And so the base peak is shifted by 16 relative to the tropylium ion. And there's a variety of other examples like that. Where you do not see the tropylium ion or its analog is when you have either a primary or secondary amine, so N-alkylated cathinones, or when the alkyl chain out here is much shorter. And so an example, if you just have a methyl, an N-methyl cathinone, then you do not see it. And in a variety of other situations, if the R group here is too short, then you don't see the substituted tropylium ion. And just to show you some examples in spectra here of a variety of different cathinones, in this particular example, you have a primary N-alkylated cathinone with a longer chain length, and you do see the tropylium ion, but it's not as abundant as it would be with a pyrrolidine substituted cathinone. Here is a very short chain length out here and a methyl alkylated N-alkylated cathinone, and you do not see the substituted, in this case it's methyl tropylium ion. You don't see it there. Again, with a short alkyl chain, you don't see it. Short alkyl chain, you don't see it. Short alkyl chain, you don't see it. In this particular case, we have a long alkyl chain. It's methylene dioxy substituted, and you do see it. It's M over Z135. Not terribly abundant, but you do see it. And again, this is basically because this methyl group is not such a good leaving group as the pyrrolidine ring. So we conducted some experiments in our lab to see if we could elucidate the pathway of how we get to the tropylium ion. So here's just looking at alpha PVP, protonated in a linear ion trap, just doing tandem mass spectrometry. That's the tandem mass spectrum. You can see at lower collision energies, we just see the loss of the pyrrolidine ring, and you can see the tropylium ion here, M over Z91. And at higher collision energies, we could basically have less abundant fragments at 161 and more abundant at 91. What's shown in the bottom left is an MS to the 3 spectrum. So in this case, we took the precursor ions at M over Z232, fragmented them, and then isolated the intermediate at 163, then fragmented that. And what you can see is that we've got a distribution of product ions, one of which is the tropylium. And this tells us that at least it's a possibility that M over Z161 is an intermediate for 91 in this spectrum. And of course, we would have to do this for all the possible intermediates that are between the mass to charge ratio of the tropylium ion and the precursor to elucidate all the potential intermediates. And it's almost impossible to tell exactly what the kind of the flow is through each of these intermediates to a low mass product unless you can do what's called a double resonance experiment in these ion traps. It's quite a complicated experiment, but there is a way, but most commercial instruments don't let you excite 2 different MOZ ions at the same time, so we can't do it on this instrument. Just a note here, I am showing this intermediate structure, MOZ119, as a phthalate type ion. And the reason I'm doing that is because we have quite significant evidence that that is the actual structure. And I will get to that in a little while. This is an MS to the 4 spectrum. So in this case, we took the precursor at 232, fragmented it, isolated an intermediate at 161, fragmented it, isolated the MS to the 3 product at 119 and fragmented it. And you can see that it fragments exclusively into the tropylium ion at MOZ91. And that's to say then you can have a variety of sequential neutral losses before you arrive at the tropylium ion. And there are many other pathways that do get you to the 91, but this tends to be what we think is the major one. We then, with our collaborators at Auburn University, Randall Clark's group, they synthesized some isotope labeled compounds. And so in this particular case, we have the C13 in the carbonyl carbon position. So the precursor shifts by 1 from MOZ 232 to 233, as you would expect, because the carbon has an extra neutron. And instead of this intermediate being at 161, it's now at 162, and that's because, again, it contains that C13. So anything that contains the C13 now is shifted by 1, and in cases where the C13 is lost, it shows up at the normal mass, as it would for unlabeled alpha PVP. And so what you recognize immediately in the spectrum then, hopefully, is that this fragment at MOZ 91 is at 91. So it's still the tropylium ion, C7H7, does not contain the C13. There's no evidence that it contains it at all. There's no 92. So that tells us then that this carbonyl carbon does not show up in the tropylium ion. It's completely absent. And we can go through a variety of intermediates. Here we're isolating the 162, which is the base peak in the tandem mass spectrum, fragment it, and you still see the 91. And this is true for all of the different fragmentation pathways we went through. And just showing on the right-hand side here, even if we take the 120, which corresponds to the MOZ 119 before, you still get exclusively MOZ 91. So even though this intermediate at 120, which is this small phthalate type ion without the methyl group, even though that does contain the C13, by the time you kick out the carbonyl, the CO group, you no longer contain the C13. So there's no way, just to say one more time, there's no way to get the carbonyl carbon onto the tropylium ion. It does not happen. Here's an example of C13 labeling on the alpha carbon of alpha PVP. So this is the carbon that's kind of in between the carbonyl carbon and the nitrogen on the pyrrolidine ring. In this particular case, again, everything shifts by 1 that contains the C13. And now what's interesting is in the MS to the 2 spectrum, the tropylium ion almost exclusively contains that C13. So that's to say then that the alpha carbon is the one that ends up in the tropylium ion. This is just showing that everything that we expect then is quite consistent. Here's showing a intermediate fragment at MOZ 134 that contains the carbonyl carbon, sorry, the alpha carbon that's labeled, and it's the one that ends up in the tropylium ion. And again, down here, even if you isolate the MOZ 119, that has the C13 in that alpha carbon position, so it shows up at MOZ 120, then that's the carbon that almost exclusively ends up in the tropylium ion. So this is basically very convincing evidence that the carbonyl carbon never ends up in the tropylium ion, and the alpha carbon almost always ends up in the tropylium ion. Just some corroborating results. Here we have a O18 label on the carbonyl oxygen, and basically everything shows up exactly where you would expect. In this case, most of the product ions are shifted by 2 because they contain the 2 extra neutrons of the oxygen 18. What used to be MOZ 131 is shifted to 33, and the tropylium ion shows up exactly where it should because it doesn't contain oxygen at all. We also looked at per deuteration on the alkyl chain. In this case, the abundant fragment that used to be at MOZ 161 shifts by 7 because there are 7 deuteriums on this particular fragment. And what's interesting now is that there's a distribution of tropylium ions, mostly at MOZ 92, 93, showing us that there are either 1 or 2 deuteriums on that aromatic product ion. And so you can imagine an intermediate here, for example, that's shifted by 1 relative to the MOZ 119, so it contains 1 deuterium. You can see the distribution of intermediates that contain 1 or 2 deuteriums, and then the final tropylium ion contains 1 or 2 as well. And ultimately, what we showed in this publication is that as long as you have a good leaving group like this pyrrolidine ring, and as long as you have enough of an alkyl chain, you very readily form the this product ion that corresponds to the loss of the pyrrolidine ring. That's the most abundant intermediate at low collision energies. And at higher collision energies, it continues to spit out neutrals through phthalate-like intermediates that ultimately end up at the tropylium ion. And we've elucidated the probability or the distribution, if you will, of at each of these levels of tandem mass spectrometry that finally arrived at the tropylium ion. And of course, the ultimate relationship between the tropylium ion and the precursor is just determined by the collision energy that you choose for your particular conditions. Regarding the structure of the phthalate intermediate, we collaborated with a group, Victor Rizov's group, and they conducted a variety of calculations to calculate the lowest energy conformers, and also ion spectroscopy experiments with Jos Uman's group in the Netherlands. And the short story here is that the lowest energy conformation is the phthalate type ring, double ring intermediate. And the charge here is quite fixed. It doesn't move around, even though the molecule looks symmetrical. There's two hydrogens down here, one up there, and there's a barrier for that hydrogen transfer. But either way, that ether shows up very much in the ion spectroscopy. And if you have any other structure at all, whether it's a ketone or aldehyde or anything like that, then the stretching frequency shifts to a place that is not observed and not supported by the ion spectroscopy at all. It's a way too long a wave number. So basically, the only ion structures that are consistent are those that contain an ether type structure, basically is a phthalate type intermediate. We also conducted similar experiments on kind of the analog that has an extra methyl group. So it shows up at MOVZ133. The lowest conformer, again, is the phthalate type structure relative to anything else. In this case, we have an epoxy type ion. And again, you see that the absorption spectra that are observed of those ions in the ion trap are more consistent with a phthalate type structure than they are with anything else. And I've just shown one example of each of them. So this gives us a great deal of confidence that the structure of those intermediates are those phthalate type ions. And so in these publications, we've just shown a couple of pathways just to propose mechanisms for how you get there. Almost all the pathways are previously demonstrated in the literature. They're usually just four center rearrangements, relatively simple. The one thing that we had not anticipated that is observed for sure is that this carbonyl group, and we're not sure that this carbon here, the alpha carbon, ends up on that particular carbon on the ring, or if it's another carbon on the ring. But for sure, the atoms that are left behind are consistent with this mechanism, whether or not they're attached in that particular order. So basically, you can go directly from MOVZ162 with the C13 label in that carbonyl carbon position and end up with MOVZ133. So just the loss of the carbonyl group, definitely observed. We observed that with high mass accuracy. And then a variety of other fragmentation pathways that basically show how you can get to the phthalate type intermediates. This just shows you that when you have the isotope label in the different positions, so in this case, this is when you originally have the carbonyl carbon and the C13 is up here with the charge, versus when you have the alpha C13 carbon, that you can follow exactly the same mechanism and end up either with the C13 retained in the tropylium ion, as you do with the alpha carbon C13, versus the C13 not being retained, as is the case for the carbonyl carbon. We just demonstrated as well that these mechanisms are conserved even when you have functionalized aromatic rings, in this case, a 3,4-methylene dioxy. So again, the precursor had pyrrolidine ring. In this case, it was perduterated, and that was lost, and there's no evidence that those deuteriumins end up in the prototyne spectrum at all. So that's the first part of the study. The next part of the study was looking at a variety of other alpha pyrrolidinophenone structures. And this just again supported the previous results that just showed that pretty much whatever the substitution is on the aromatic ring, the base peak in the prototyne spectrum is the functionalized tropylium ion version of that. And there really weren't any surprises in here. What we did learn is that the preferred pathway is first the loss of the pyrrolidine ring to a kind of phthalate type intermediate, and then to the tropylium type ion. And the higher the collision energy, the more you form the tropylium ion, and the lower the collision energy, the more you stay behind some of these intermediates. And the only other kind of general trend really is that the longer the alkyl chain, the more variety of intermediates you saw. So for example, the MOVZ133, and you'd see 14 Daltons or extra CH2s above MOVZ133. So for example, 147 and 161, things like that. And this is just showing that we also did look at the electron ionization mass spectra, as one would see in traditional GC mass spec of all of these substances. And basically, there weren't any surprises. The tropylium ion is far less abundant here. In fact, practically non-existent in most of these spectra. And that's because the other pathways are so kind of predominantly preferred. And this is very well known in the literature. So this is just to say that there are really no surprises. And when we look at the EI mass spectra, the C13s do exactly what you'd expect. Okay, in terms of the N-alkylated cathinones, we looked at secondary amines with just one N-alkylation and tertiary amines with NN dialkylation. Here's an example of one that is has a D3 group here. Whether we also looked at electrospray ionization, as one would see with HPLC, QTOF, for example, or HPLC ion trap mass spectrometry. We also looked at DART because we know that some labs, such as at the Virginia Department of Forensic Science, are starting to do DART MS or DART MSMS on some of their casework as screening. The long and short of this is that the tandem mass spectra from DART were very similar to the ESI tandem mass spectra, with a few exceptions, and that is that DART mass spectra tended to have a little bit more noise, and they tended to prefer to have higher energy fragmentation pathways, which sometimes led to more neutral losses, but always led to higher abundant low mass ions. So again, DART mass spectra tended to have more abundant low mass ions, and traditional ESI tandem mass spectra tended to have less abundant low mass ions. So kind of the intermediates are more observed. Some common observations here for the N-alkylated is that when you have secondary amines, so basically one R group on this amine, then you tended to have a loss of water. That was the preferred pathway. And then you may also see radical losses. So even though the precursor was just protonated, it was an even electron precursor, you definitely do see evidence for radical losses, so odd mass losses. So you can go from an even mass precursor to an odd mass fragment ion. We are certainly not the first to observe this at all. Amelia Fornell's group has shown several examples of this, and several other groups have as well. And it's one of the few groups that I'm aware of in terms of groups of compounds that do produce radical ions from even electron precursors at relatively low collision energies. And so in terms of spectral interpretation, this is going to be quite difficult for practitioners of all kinds, because this class of molecules behaves so oddly relative to other classes of drugs. And again, the main point is you do see radical losses from an even electron precursor. So just to sum up all the work we did on cathinones then, origin of the tropylium ion was identified. We've got the pathways. We've used isotope labeling to figure it out. We've discovered the structure of some of those intermediates. They're consistent with a phthalate type intermediate. N-pyrrolidine type cathinones follow very conserved fragmentation patterns. They lose the pyrrolidine first, and then they have a sequence of neutral losses. N-alkylated tend to lose water if you have secondary amines, and they tend to form alkylphenones if you have tertiary amines. And again, you can see abundant radical losses from secondary and tertiary cathinones, but you almost never see radical losses from pyrrolidine cathinones. All right, moving on to fentalogs. I don't need to spend much time talking about fentalogs other than to show you that, and everyone's aware of this, that it's a major problem in terms of addiction and overdoses, accidental overdoses right now. There's a very wide variety of fentalogs that have appeared in casework, and you can have functionalization in a variety of different R group positions here, as has been very well described in a variety of places. So we did a variety of mass spectrometric studies. We looked at GC-EI mass spec, and we also looked at HPLC, ESI, MS, and a variety of other groups have studied this as well. For example, Jennifer Millett and John Casale at the DEA showed the difficulty in telling the difference between 2-methyl and 3-methyl fentanyl, or cyclopropyl and crotonal fentanyl. Ota's group showed that a lot of the fragmentation pathways are conserved even when you functionalize in different places. And of course, the NIST group, Morphy et al., have developed a hybrid similarity search, which is based honestly on the conserved fragmentation pathways that tend to be observed. And this hybrid spectral similarity search uses a variety of product ions and neutral losses from the precursor and uses them together to try and give you an idea of where the R group substitutions are occurring in a given spectrum. So in terms of protonated tandem mass spectra, again, some of the fragmentation patterns can be very similar between these different fentanyl structural analogs. It's really hard to tell them apart. So if we can use fragmentation patterns to help figure it out. That would be really helpful. Whitney-Had's group, this is also at West Virginia University. This is Pat Keller's group, showed that there are two major intermediates at MOVZ 188. I want to say, or intermediates or product ions, MOVZ 188 for fentanyl. And this is in the protonated tandem mass spectra. So, for example, in a QTOF. And they did this with isotope labeling and showed that you can either have like an unsaturated alkene, kind of cycloalkene up here, or not. And so the way they established this is both with isotope labeling and then tandem mass spectrometry, so MS to the 3. And when you take this particular fragment and fragment it, you pretty much exclusively get MOVZ 135, which of course without the deuterium would show up at MOVZ 134. And this particular form gave a variety of product ions. When we studied fentanyl, protonated fentanyl in our lab, we saw similar observations. So the precursor here is at 337. We've shown the structures of some of the intermediates here, or some of the product ions. This product ion at MOVZ 188, again, the accurate mass shows that it's consistent with elemental composition, and the two structures from the Whitney-Had and Calorie group is shown there. And again, we just did MS to the N to fully flush out all the possible pathways that exist. And this is just showing the two different intermediates and how you can get there. You either have kind of a hydrogen shift over here or not in the process. In one case, you can have a retro-Diels-Alder reaction that leads to 134, and this tends to be the preferred pathway here. Or over here, you can have a hydrogen transfer and then kind of a contraction or the elimination of a butadiene that gives you the 105, or a contraction the other way. To spit out a cyclopropene group to give you the structure down here. So again, nothing really surprising relative to the literature that already exists on those. In terms of MS to the N, again, we kind of dug down in these pathways. We figured out, just flushed out more of these mechanisms basically, and we used some isotope labeling with high mass accuracy as well to flush some of this out. I'm going a little bit fast here just in the interest of time, but if you want more details on the explanations of exactly what's going on here, they are in the publication that's cited at the bottom. And again, in most of these pathways, we try to use the simplest possible mechanisms to explain the observations, and they tended to be 4-atom cyclization elimination reactions. High resolution mass spectrometry of that precursor, or the intermediate at 188, again showed that high mass accuracy is consistent with that particular elemental composition. However, you can see in the top here, there is evidence of another intermediate or product ion at MOVZ 188.1075. It's measured here at 1086, so the less than 5 parts per million mass accuracy. And that elemental composition is consistent with what's shown here on the top right. So basically, it's very low abundance, but it is there. And so really there's a third structure, has a different elemental composition than the other two conformers, and this conformer has the arrangement shown up here. We also identified a novel R group transfer in the fragmentation pathways of fentanyl, and this was not previously described in the literature. So what we're looking at here is a fragment that has this elemental composition based on the high mass accuracy. And we believe it has the structure shown just because of the mechanism. And so if you draw fentanyl in this boat conformation here on the central ring, then it's possible for to have nucleophilic attack from the lone pair on this nitrogen to attack the carbonyl carbon and basically cleave the aniline or the amide carbon-nitrogen bond and move that R group, that R2 group, to this nitrogen position over here. Once that R group transfer occurs, you can then have a simple 4-center rearrangement to spit out the aniline ring, which would then give you the intermediate at MOVZ 244. That can then rearrange or spit out some neutrals via a variety of mechanisms to either give you MOVZ 190, which is the most abundant fragment in the fragmentation of the intermediate at 244, or it can give you 188, which has the same elemental composition as the two conformers identified by the Whitney-Had group. And this can, of course, rearrange to spit out to give you 134. What we showed here is that if you, that's shown in the bottom of this figure on the left-hand side, is that MS to the 4, so shows that you form either MOVZ 134 or 120 from that intermediate 188. And that just shows you then that this fragmentation pathway is a little bit different than the fragmentation pathway from the other two structures that Whitney-Had et al. Isolated. So really, perhaps a fourth structure in there somewhere. This is just showing the high resolution of MOVZ 190, just to show that we do have the elemental composition shown below. And this is just showing that we did study a variety of other isotope-labeled fentanyl analogs, and they were all consistent with the mechanisms and the pathways that we've shown in these publications. And that's just to say that when you have, for example, per-deuteration on the aniline ring, they all leave and none stay behind, and there's really no surprises there, which is good. In terms of R group modifications, this is really of interest when you're trying to identify new novel psychoactive substances, or fentanyl analogs, I should say, sorry. What we really showed is that when you have R1 and R2 substitutions, there's no surprises, but these less abundant fragments just move around a little bit, depending on exactly where the R group substitution is and exactly what you'd expect them to be. So that's to say, really, that the actual identity or the fragment assignments for these particular fragments are very useful if they happen to move in a given spectrum, because then you can tell what R group may have been added to that particular part of the molecule. And this is really taken advantage of in the spectral similarity, like the hybrid search that NIST is developing. This shows that when you have R3 and R4 substitutions, so R3 will be on the central ring here, and R4 will be on the alkyl linkage between this particular nitrogen and the whatever aromatic substitution is on the end down here. R3 and R4 really radically change prototyne spectra more than substitutions anywhere else. So they basically tend to give a wide variety of peaks and also shift the abundances to kind of somewhat unpredictable places. And so, if you're quite sure that you have a fentanyl analog, and if the tandem mass spectrum shifts very dramatically, not just by the movement of a few low abundance peaks, but by radical changes in the spectrum, you probably have an R3 or an R4 substitution. R5 groups tend not to change the spectrum very much at all. They follow conserved pathways. This is just trying to summarize the influence of the different modifications and the pathways they tend to enhance. So for example, if you have R3 or R4 substitutions, they tend to prevent the R group transfer that's observed. And they tend to favor the loss of the R group. So for example, when you have a substitution on the R3 group, you tend to lose the R3 group if it's a good leaving group, like an OH group, or a methoxy or something like that. And so you might form something like this. And finally, if you have a bulky R1 or group or R2 group, then they tend to just follow those particular pathways. So in summary, we looked at a variety of fragmentation mechanisms. We found a third nominal isobar, M over Z 188, and possibly a fourth structure, M over Z 188, that goes through a different pathway entirely. And the three main isobars are shown here. We identified an R2 group transfer that was not in the literature before. This R2 group transfer tends to be enhanced when you have a good amide kind of transfer group, and tends to be blocked when you have a bulky group on the kind of the central ring that kind of prevents that boat conformation and the R group transfer totally. We studied R group effects and basically established that if you have R3 and R4 groups, they tend to be good leaving groups, and direct pathways in quite unique ways relative to other fentanyl analogs. So finally, to the last part of the talk, the spectral identification and the development of a new algorithm. It first helps to look at how current algorithms work a little bit before we tell you how our new one works, or what we're proposing. Here's an example of a NIST cocaine spectrum that we have basically done a NIST search on, and excluding its own identity as a possible identity. There are some replicate spectra of cocaine from other laboratories in the NIST database, and it's found one of them as the next best hit. And so this is just showing basically two replicate spectra of cocaine conducted in different labs. And this should be of no surprise to anyone, is that the relative abundance in these electron ionization mass spectra is a little bit different. And we know that these abundances tend to shift a little bit if we look at replicate mass spectra, even across a GC peak on the same instrument collected almost simultaneously. So we know there's some random variance in abundances. And what's interesting in this particular case is the match scores and the probabilities of those identities that comes out of the NIST algorithm. And the NIST algorithm, by the way, just calculates a weighted dot product, or kind of an angle between the two spectra. It's a lot like a Pearson product moment correlation between every ion abundance across the spectrum. It just weights them. It gives a little bit more weight to high mass ions, and it just weights the intensities to enhance probability that the correct identity will rank higher in the top 10, the search list results. What's interesting here is if we send NIST to the NIST database, it comes back with an 88% probability of being cocaine, but an 11% probability of being pseudococaine, which is only different by the chirality on this particular carbon up here. Cocaine and pseudococaine only differ in that position. There's also a 1% probability of being allo-pseudococaine, where the R group, the chirality is different over here, and a 0.25% probability of being allococaine. And so basically, there's a 99% probability of being some form of cocaine, but can't quite elucidate what the stereochemistry is. And in some cases, especially if you have a chiral column, you can separate out in retention time these different isomers, these stereoisomers from one another quite easily. But the point is, can mass spectrometer do better all by itself? Like, do you have to have the GC to help make that identification? Or can we use the fragmentation pattern a little bit better than we have been? And so just to show you what we're thinking of in terms of this new algorithm, I'm going to compare the way things are done right now, which is what we're going to call like having a static exemplar. And when I say static, we mean that we assume that there's one good spectrum of cocaine, like one true spectrum, one accepted spectrum. And this is true for all drugs. And what does the good one look like? Like, what is that static good one that we're going to compare things to? And these exemplars are what exist in the databases. So for that purpose, let's just consider 3 fragment ions and what might happen to 3 fragment ions in replicate spectra, right? So these might be 6 replicate spectra across a GC peak. And so, for example, there'll be some variance. And what we could do is average across a GC peak, or just look at the apex, the most abundant spectrum across the GC peak, and we might see something like this. And in this particular case, let me tell you that this average is literally the average of these 6 spectra over here. It's not pretend, it's literally the average. The confidence intervals here are literally the confidence intervals that were calculated from these 6 replicate spectra. So here we go. This is the average of those 6, and these are the 6 individual ones. The way that current algorithms work is they basically take the exemplar and the known uncertainty, and they say, how well does a question spectrum match the exemplar, given the tolerance or the uncertainty that we have in our measurements? And in a situation like this, you would, is the question spectrum significantly different from the exemplar? And in this particular case, you'd have to say, well, no, it's not different, because the ion abundances of these 3 peaks all fall within the 95% confidence interval of our exemplar, and so there's no significant difference. The exemplar has to be a possible identity for the question spectrum. And this will be true for all of the exemplars for which the question spectrum fit within. That's where we currently stand. That's basically how the current algorithms work. They all have an exemplar, a fixed exemplar, with uncertainty, and they compare question spectra to it. What we're proposing is a little bit different. And what we're saying now is, does the question spectrum have different behavior than the standard, like dynamic behavior? And if you look at the abundances of the gold and the blue peaks, what do you notice in all of the spectra? Well, in the first spectrum, they only differ by 1 relative to one another. In the second spectrum, they don't differ at all. And in all of the other spectra, they only differ by 1. Sometimes the gold is more abundant, sometimes the blue is more abundant. But the point is they correlate very closely. So when one goes up, the other goes up. When one goes down, the other goes down. And if you look at the behavior in the question spectrum, you can see that there's rather a large difference between those two. And you'd have to say, you know what, based on the pattern of behavior, this actually looks significantly different than the behavior that's observed for the 6 replicates. And so you would have to say there is reason to think this is significantly different. Another way of saying this is, if you couldn't see the abundance of the gold one down here, what would you predict it would be? And based on the pattern of behavior for these 6, you'd have to predict that the gold would be up here. It's not, it's down there, and that's a large residual or a large error, and that's a reason to exclude it as a possible identity. And this is what our algorithm does. It basically pretends that we don't know an abundance in the question spectrum. We then use the pattern of behavior in the replicate spectra that we do have to predict the ion abundance in the question spectrum. We then compare the predicted ion abundance to the measured ion abundance at every m over z that we've predicted, and see how well we've done. So let's just show you how to do that then. The fragmentation pattern of cocaine, by the way, this is one of the reasons we chose it, is because the fragmentation pattern is so well known. It was very studied in detail by Smith and Casale here in the 2010. And the fragmentation pathways in the EI spectra look like this. And you might expect then, or predict, what about two fragments like this? If the 77 only derives from 105, do they correlate? Do the ion abundances and replicate spectra correlate, or don't they? And here are 135 cocaine spectra collected from a crime lab over the period of 4 months. This is a cocaine standard that was run. It's not casework, it's a standard that was run over 4 months. And you can see that they do correlate. They correlate with an R squared of 0.68, which tells you that you can predict the abundance of 77 with about 68% accuracy if you know the abundance of M over Z 105. And let me just remind you the way that this algorithm works relative to other algorithms. So static algorithms like the NIST one assume that there is one exemplar that's the good spectrum of cocaine, and that's based on the mean, basically, of a GCP, or the mean of spectra. In this case, if we look at the mean of the database, the mean of the abundance of 77 is 30.9. The mean of the database of M over Z 105 is 30. And so if you have any one spectrum, if you had to predict the abundance, you would predict it based on the mean. However, in any one spectrum, if you actually knew the abundance of M over Z 105, and let's pretend it's 24% of the base peak, then I think you'd be insane to predict the abundance of 77 to be the mean of the database. You'd be far better off using this correlation to predict the abundance of M over Z 77 from the abundance of 105. And that's to say that M over Z 105 is a better predictor of the abundance of 77, not the base peak. These abundances are relative to the base peak, whereas this correlation is relative to one another. So again, another way of saying this is that if the relative abundance of 105 is 24% in a spectrum, then you would predict like 23% for M over Z 77. Now this is a binary correlation, so it's predicting one dependent variable from one independent variable. So one ion abundance from one ion abundance. But you might think that you might do better off if you actually predicted one ion abundance somehow lots of times, or from different ions with which it correlates. And that would be a multivariate approach. So you predict one dependent variable, one ion abundance, from many ion abundances. And that's exactly how our algorithm works. We use a general linear regression model, multiple linear regression. We predict an abundance of one particular ion in the spectrum based on the other ion abundances. And we just need to figure out the coefficients that go before each of those ion abundances, these different B terms, to be able to predict an ion abundance. And what's shown here is just the output of one of the models that we've calculated. Here, the dependent variable, the ion abundance we're trying to calculate, is M over Z 82. And here we're using maybe 14 different product ions, and the 14 most abundant product ions in cocaine, to predict the abundance of 82. B is the coefficients of each of them, and kind of this partial eta squared tells you really how effective each of those coefficients are, or each of those mass to charge ratios are, in predicting the abundance of 82. So when you have a very small partial eta squared, like 0.001, and a very small coefficient, then this really is not very helpful for predicting the abundance of 82. But the larger the partial eta squared, or the observed power, for example, here M over Z 42, you can see has a high observed power, or one of the larger partial eta squares, and it has a much larger coefficient, so it's more useful for predicting the abundance of 82. So we can use this model in a variety of ways. We can either leave all of these coefficients in and all of the M of Z's, or we can use a model to say, you know what, just include however many fragments are necessary to explain as much of the variance as possible, and then don't use any more. And if we allow that to happen, then the model usually lands on 4 or 5 ion abundances to predict the abundance of any one dependent variable. Or we can use them all. And our work is basically in the process of showing that it doesn't really matter. You end up with really good predictions either way. This is just showing again that there are some fragment ions that are not useful for the predictions, so we can leave them out, and other ions are very useful, so we can leave them in. This bar chart or histogram is just showing the mean and the 95% confidence interval of the database spectra. In purple, the second in the series is a NIST cocaine spectrum that we have the measured ion abundances of, and this was taken from the NIST database. So this would be considered an external validator, right? It was external to the database. And what we have in red is the prediction of the ion abundances of that NIST cocaine spectrum based on the pattern of behavior or those multiple regression models of the database. Again, just reminding you that the NIST spectrum was collected on a different instrument 30 years ago. And what you can see in some cases, let's just look at M over Z 182, the NIST cocaine spectrum falls a little bit outside of the 95% confidence interval, and it's it's a good almost 20%, maybe 18% away from the mean of the database. Whereas our prediction, our model that's based on the database, predicts it's down here at like 65% when it's actually at 72%. So we're closer, we're not perfect, but we're closer than the mean of the database. And in most of the cases you look at down here, let's look at M over Z77, you can see that the NIST cocaine abundance is about 4 or 5% larger than the mean of the database, but our prediction is right on, it's a fraction of a percent difference. And at most of these M over Z values, where the ion abundance differs from the mean of the database, our model is a little bit better. There's a couple of cases where it's not. But in general, the prediction is better. Here's just showing a correlation between the measured abundance in that external validator, that NIST cocaine spectrum, relative to the mean of the database. And so you can see there are some quite large residuals or large errors, and the correlation between the two is only, or the R squared is only 0.94. Whereas with our model, this is the NIST ion abundances again, and then our general linear model predictions for those ion abundances based on the database. And you can see that our residuals are a little bit smaller, at least visually, and that the R squared is superior. In other words, it's the Pearson product correlation is better for our model. Just showing you a non-match. So here we're looking at pseudococaine relative to the cocaine model. Again, this is a known negative, and so we expect them to be significantly different. What you can see is that there's one case down here, for example, M over Z94 is less abundant than in pseudococaine than you would expect for cocaine. And indeed, it's different from the mean of the database, and our model also predicts it should be up here if it was cocaine. Well, it's down there, so it must not be cocaine, right? And there's a few other cases as well. Here's an interesting one where M over Z77, where the ion abundance is within of pseudococaine, is within the 95% confidence interval of the cocaine database, but our prediction puts it way down here. So just an example where our prediction has a larger residual for the known negative than the database for the known negative. So try to summarize some of these quantities that we're calculating. We can either look at the residuals themselves and the kind of magnitude of those residuals with multiple guesses or multiple predictions, or we can look at the Pearson product number correlations between predicted ion abundances and measured ion abundances. So we're now looking at cocaine spectra from different sources. Lab 2 in this particular case was used as the model, and the mean spectral ion abundance across 19 peaks in 132 spectra, the mean deviation is 3.1%. And for the static exemplar model, so a given spectrum will deviate 3% in an ion abundance relative to the mean of the database. And when we use our model, the general linear regression model, that deviation is only 0.7%. From a measured ion abundance to the predicted ion abundance, which is 3 or 4 times smaller variance. That's to say, basically, that much of the variance in replicate spectra is explainable. It's explainable through this model, and it's never been explainable before. The result is that if you looked at the range of the spectral means within that data set, they vary from 1.1 to 8.2. So that's just to say that some spectra are closer to the mean of the database, other spectra are further away. And when we use our model, they pretty much all tighten up. When we use 126 cocaine spectra from a second lab, these are external validators. They're known positives, but they weren't used to generate the model. You can see that the static model that's currently used, the mean of the database, is a worse prediction in this case, but our model still does very well. It's a little bit worse in terms of the mean of a set, but the range of spectral means really isn't too bad. We've also looked at a variety of cocaine spectra. These are also known positives that are external to the database. And again, the static model doesn't do so well in terms of the mean absolute residuals, but our model still does pretty well. We also have a bunch of known negatives either collected in the same lab or just different labs, and some of these known negatives are really similar structures. They're stereoisomers, so they have the same constitutional arrangement of atoms, just different chiral centers. And in these cases, you can see that the residuals basically fall within some of the cocaine spectra. So that they're not necessarily significantly worse hits. It's not easy to tell if they're negative. You know what I mean? The abundances are too close. They fall within the observed cocaine abundances. Whereas if you look at the mean absolute residuals down here, the smallest residual for our model is 3.8% off. And what you'll notice is that if you look at the larger residual for all the known positives, it's 2.7%. And what this shows then is that in theory, you could have a threshold for a decision like 3%. And if you chose 3% as a threshold, all of the cocaine spectra, every single one, whether it was the internal database spectrum or the external validators, that they all have residuals that are less than 3%. Whereas every single example we had of something that wasn't cocaine had a residual that was larger than 3%. And just to show that kind of pictorially, this is showing the static exemplar approach, showing the mean absolute spectral residuals for 19 different predictions. Again, these predictions are based on the mean of a database. And over here is shown all the compounds that are not cocaine. So on the left is cocaine within the database on which the model is developed, and the external validators, the NIST spectra and the other lab. And on the right-hand side is all the known negatives. And what you'll notice is it's impossible to pick a threshold for a decision that is always correct. If you picked a threshold of 3%, then you'd exclude a lot of cocaine spectra as being cocaine, and you'd include non-cocaine spectra as being cocaine. So you'd have false negatives and false positives. Here is our dynamic model, and what you show is that the residuals are all less than 3% for all of the cocaine, whether they were part of the database generation model or external to it. And all of the compounds that were not cocaine, so the known negatives, all had residuals that were larger than 3%. So now we can have a threshold for a decision below which all the cocaine spectra are correctly included and none are excluded, and all of the non-cocaine spectra are correctly excluded and not included. We can use another measure of similarity between the spectra, and this is using the Pearson product moment correlations of the same data, the same database, the same predictions, the same models. And in this case, the closer to one you get, the better the similarity between the prediction and the actual measured spectrum. And what we're looking at here again is just the mean of a particular group of spectral sets. And in general, let me just draw your attention to a couple of different things. One is that the worst correlation for actual cocaine relative to the cocaine database is like 0.6. Whereas in some cases, if you look towards the bottom, you can see that pseudococaine has a spectral similarity of 0.88 relative to the cocaine database, which is a better match. That's to say, again, that pseudococaine is a better match than some of the cocaine spectra to the cocaine database. In our dynamic model, the worst we did in predicting cocaine is a spectrum that gave a correlation of 0.9879. But the best prediction that we did for something that wasn't cocaine had a correlation of 0.9746, which is worse than up there. And just to show this kind of graphically, on the top is the exemplar or the static approach. Again, the closer you are to one, the better the spectral hit. And you cannot pick a threshold below which all of the known negatives are correctly excluded and all of the known positives are correctly included. Whatever threshold you pick, you always make a few mistakes with the static approach. With our dynamic model, you can pick a threshold above which all the known positives are correctly included and all the known negatives are correctly excluded. And that's where we currently stand with the model, is that at least for cocaine and cocaine stereoisomers and all of the spectra that we've been able to obtain so far, there's kind of a good indication that this model is doing a superior job relative to existing algorithms. And in the upcoming publication that we're currently working on, we also compare it to the NIST algorithm for the actual similarity score that the NIST spectrum gives, like the weighted dot product. Well, thank you for your attention. I would be very happy to answer any questions. I will be around after this webinar. And whether you have questions about cathinone fragmentation patterns or mechanisms, or fentanyl analog fragmentation mechanisms, or the algorithm, this new approach to spectral identifications, I'd be more than happy to answer any questions. Just wanted to thank a couple of colleagues for their help and advice with developing the algorithm for spectral predictions, basically. I wanted to thank NRJ for the funding for enabling us to pursue this project, and thank you very much to RTI and the Center of Excellence here for helping us disseminate to practitioners, where hopefully we can be of some assistance. Thank you very much.
Thank you, Dr. Jackson, for such a fantastic and informative presentation. Let's get right into some questions from the audience. First one I have here is for the cathinoids and phenologues, would you expect to see the same fragment ions and pathways in DART MS as you do in tandem mass analyzers?
Yes, and the short answer is yes. Maybe if I could have a couple of examples. I guess I anticipated a question like that. Let me just share the screen for a second. Here we go. Can you see that okay?
Yep.
Okay. So yeah, here's an example of 3,4-methylene deoxy, or 3,4-MDPV. This is spectra collected on a QTOF high-resolution mass spectrometer. And so in both cases, we have the same instrument, and we either operate the instrument with an electrospray source, or we operate the instrument with a DART source. And in both cases here, we've isolated the precursor in the quadrupole. So in some ways, then the precursors have lost the memory of the ion source from which they were generated, whether it was electrospray or DART. They were isolated in the quadrupole and then fragmented and observed in the TOF. The fragmentation patterns here are very similar. You only expect them to differ by the internal energy differences that the ions have before they're excited in the collision cell. And that's to say there's very little difference whatsoever. Things are a little bit different if you do in-source CID on a DART MS instrument. And this is actually more akin to the instrument that JEOL sells. It's just their DART AccuTOF. And so there's not a way to isolate precursor ions. So what they really do is they take all the precursor ions from the ion source and fragment them all. Now, if you have a relatively pure kind of pure distribution, if that's not the correct word really, if you have like only one dominant molecular ion in the DART source, then the product ions you see will be dominated by that one. But of course, if you have 2 or 3 types of molecules all present in the precursor ion scan, then they will all be fragmenting and they will cause some kind of confusion. But in general, if you do have quite pure spectra, you might see something like this. So this is a methcathinone. This is in the top, this is in-source CID, the DART ion source, and it's fragmented in this kind of the skimmer to cone voltage difference. So you have a large voltage offset, and the ions are really accelerated in that kind of 2 Tor region as they exit the capillary and between the skimmer cone. In the bottom spectrum, it's more like beam type CID. So this is collision in the collision cell in the QTOF on the same instrument. And what you can see, there are some differences now. For example, the peaks at 103 in the top is a little bit more abundant than in the bottom. And there's some extra peaks here around MOZ 90 or 89 that are more abundant in the top than the bottom. And in general, what we see is that in-source CID, you tend to see some kinetically favored projects or higher energy fragmentation pathways. You also tend to get more noise and more low mass product ions, because when you do have neutral losses, you have more sequential neutral losses. So long story short, there are some differences, some nuanced differences between in-source CID and a DART MS and kind of true MS on an instrument that can isolate precursors before you fragment them. I hope that answers the question.
Thanks, Dr. Jackson. How do you see this algorithm being made available to practitioners? And this is kind of follow up by partnering with an MS company to incorporate it into their software programs?
Yes, yeah, whoever's willing to pay the most money is the simple answer. No, yeah, I've spoken with NIST to see if they would be interested in incorporating, because from my perspective, that's the lowest barrier. It's just like giving them the algorithm and the equations and showing them how to do it, and just having them write it in, so that when practitioners do a NIST search, in addition to getting the traditional ranking that you would get, you could then select the #1 hit up there and just ask it to add this extra layer. and then it would give you a probability that that number one rank hit is in fact the identity. From our perspective, that would be ideal. However, NIST isn't, for whatever reason, is just not terribly interested in doing that. It would take several full-time staff, a year or two to write all the code and to validate it. So that's to say we're a little bit on our own. We're limited by the amount of staff, students and postdocs I can put on this. And of course, with only one grant, it's a little bit limited. So I guess the first goal is to first get more money and then throw more students at it. And then, once we actually make more progress to try and partner with a commercial vendor, maybe Agilent would be a good one since so many labs have Agilent specs. If anyone's willing to assist with this process, by the way, I'd be more than happy to talk with you.
Fantastic. Kind of in the same vein, I've got another question here. Would the new algorithm work for MSMS spectra on tandem mass analyzers?
The short answer is yes, it will. We've tested this on ESI, MSMS spectra, and we've shown that as long as you fix the collision energy, then it's easy to have one model that that makes predictions very well. However, when you change the collision energy, you change the nature of the coefficients in the model, and so that's unfortunate. But it does mean that you need a different model for a different set of models for every collision energy that you use. Which means that if you were ever to do this in practice and use this algorithm to try and identify substances based on their tandem mass spectra, you would have to do them all but that drug would always have to be run at the same collision energy. Otherwise, you'd need a whole bunch of different models.
Thank you. Another question I have here, do you expect the algorithm to work on other classes of drugs like cathinones or fentanyls?
Oh, yes. So I'll just share the screen again just to show an example here of some work that's currently ongoing in the group. So we have extended it to fentanyls already. So I'm just trying to. If I can come down here to the bottom, I have. There we go. Is this still visible? Can you see?
No, it's okay. It's switched off. You might have to share again.
Okay.
There you go.
So here's an example where we applied the model to 4 fentanyls and 4 cathinones and 4 opiates. Again, we have a variety. This is EI mass spectra, so GC EI mass spec. And what you can see here is just showing the Pearson product moment correlations for 4 different fentanyls. And this is the average internal PPMC. So these were spectra in this particular column that were used to generate the database. So we expect to do well on them because they were used to generate the algorithm. And then we have external validators. So these were spectra that were not part of the database generating model. You can see that the Pearson product moment correlations are equally good for those as they are for the spectra in the model. And then we can also look at the root mean squared error of predictions. This is similar to the absolute residuals. And so you can see that basically, whether it's the internal validators or the external validators, equally small residuals. So whether you look at the similarity scores or the difference scores, they basically show that our predictions are really very good. And I haven't necessarily, would I show it here? Yes, and here's kind of getting towards kind of the confusion matrix table. So what we expect on the diagonal is the Pearson product moment correlations at the top for spectra with different identities relative to the models on the left-hand side. So the rows are the models and the columns are the kind of unknowns that we're comparing to the model. On the diagonals, of course, the model is trying to predict the actual identity, and so we expect it to do well on the diagonals, and we do. And off the diagonal, you would expect it to do bad, and you can see that the Pearson product moment correlations are significantly worse off the diagonal, as they should be, because these drugs are the wrong identities. At the bottom is all the same predictions, but just compared a different way. We're looking at the root mean squared error of predictions. The diagonals have the smallest root mean squared error of predictions because they are the correct identities, and off the diagonal, our predictions are worse. And so in all of these cases, you can imagine having a threshold for a decision. And in every case we've found so far, that threshold makes very few or no mistakes for as many spectra that we've looked at. And so yes, the model will work for other drugs, and it's just a matter of collecting enough replicate spectra of enough drugs to be able to build enough models to be able to basically identify any scheduled drug.
Another question that I have here, what are your requirements for students that want to help you with your research?
That's a very good question. So basically, it would be the admission requirements for the university. So we have most of the students that contribute are either master's students or PhD students, and they would just need to meet admission requirements. So that would be a GRE, a GPA above a 3.0 out of 4, preferably some research experience, and just passing the interviews. And that would be the criteria for admission. And then once they're in the program, I'd be more than willing to consider having them join the group.
Thank you. I think we have one more question here. For the fentanyl study, how many different structures did you consider at the M/Z119, and how relatable are the DFT and spectroscopic measures?
Ooh, very good. So, gosh, we looked at probably 20 or 30 fentalogs that went through that particular pathway, and they were all the ones we checked were consistent. I think we provide a list of the names of those fentalogs in the publications, and a lot more spectra are in the supplemental material. And can you remind me of the second part to that question, please?
How reliable are the DFT and spectroscopic measurements?
Yeah, so in general, those measurements are very reliable. I think I have some examples here of some of the additional. Here we go. Just share this quickly. Share screen. Okay, so what we're looking at here is a variety of different density functional theory calculations to try and assess the minimum energy structure. And this, just because these are the minimum energies, doesn't mean that the molecule can always rearrange to get there. There may be a barrier to getting there, but if it can get there, then these are the lowest energy structures. Basically, what's shown is, of the 7 or 8 that we considered as reasonable, that the phthalate type intermediate is by far the lowest energy. In this particular case, it's almost 140 kilojoules per mole lower energy than the nearest structure, which is this epoxide ion. There's no barrier, by the way, of conversion between the epoxide and the structure up here. So, I mean, the uncertainty on these calculations is on the order of, I don't know, 1 or 2 kilojoules per mole. They're extremely reliable. And then in terms of the predicted spectra, the wave number accuracy is extremely reliable in these calculations, usually by a few wave numbers or a few fractions of tens of wave numbers. The abundance is very difficult to predict a priori, which means that you really can't trust the abundances very much relative to one another, and that's due to all kinds of reasons with the ion beam intensities and spectral transmission and all kinds of other factors. But you can definitely trust the wave number accuracy. And the fact that this phthalate ether type moiety lines up so well in this particular case is extremely reliable. There's really no possibility that it's a carbonyl group in the intermediate. The fact that we haven't hit all of these stretching frequencies means that there's probably other structures in there, but we haven't identified those yet.
I actually had one more question come in, if we want to answer that real quick.
Sure.
How well does this algorithm perform against real patient data with interferences on the MS2 spectra?
So that's an interesting question. So yeah, when the rubber hits the road, how well does it do on casework? So the real problem there is we would never know, and the reason is with casework, you don't know ground truth. So you don't know if the drug is there or not. You can only really assess the error rates from known positives and known negatives. So you can only really do it with standards. Of course, it's possible to take body fluids, like whether it's a urine or blood or plasma, whatever it is you're analyzing, and spike it with a known, and then see if you can identify it. That would work. We haven't done that yet. And so, yes, for sure, additional noise on those peaks will contribute to uncertainty or variance, and our model will do worse. But so would any other model. And the fact that we start off superior, I think, would mean that we would end up superior to other models. So I guess that's an optimistic take, but I hope that answers the question. I'll try to be as honest as possible with that. We will do worse, but so would any other algorithm do worse.
All right. Well, thank you. I think that makes all the questions for today. So we'll go ahead and close out today's webinar. So I'd first like to thank our subject matter expert for taking the time out of their day to present the material for today's webinar. So thank you again, Dr. Jackson, for taking the time to provide such a wonderful presentation.
Thank you very much, and thank you for the opportunity to share our work with as many people as possible. I appreciate it.